Just 1 second. Well, good evening, everyone. Good evening, everyone. That's when, good evening. Such a great crowd. Such a great crowd. We are so excited. So I'm going to be brief because everybody wants to know what is the BRAIN Initiative. My name is Christina Eleki, Executive Director for your host organization, the Chicago Council on Science and Technology. We are a nonprofit organization, and we seek to enhance the public's understanding of science and technology and its impact on society. We were founded in 2006, so next year we're going to celebrate our 10-year anniversary, and I would like to point out our president, Dr. Ellen Schriesheim, who is sitting down there. Ellen, can you please wave your hand or stand up? Ellen is the president since day one, and he's also director of emeritus of Argonne National Laboratory. I would like to thank the Chicago Community Trust. This program tonight was made possible by a grant provided by the Chicago Community Trust. So thank you, Chicago Community Trust. Argonne and the Neuroscience Society of Chicago chapter, we partnered, and thank you for your partnership. Argonne National Laboratory, thank you. Neuroscience Society Chicago chapter, thank you so much for your partnership. I wanted to mention that we are a membership-based organization, and your support as a member, it's a great help. If you're not ready to sign up, you can also provide smaller donations, so please go to our website. If you picked up this little sheet, you have other ways to get involved to the organization. We have a Facebook page, a Twitter, a LinkedIn account, and a YouTube channel. We record all our programs, and actually there is a live streaming tonight too. Check out the programs that you missed before. And I want to mention upcoming programs. We have a great program lineup for the fall, but I'm going to highlight 3 programs. Why Science is so successful is going to be a book review by Stuart Firestein, and it's going to take place at the Chicago Tech Academy, October 21st. November 12, A Picture is Worth 1,000 Words, Teaching Science with Comics. It's going to be a workshop with the comic nurse. And in December, Women in STEM. Women in STEM is our signature program. We're going to feature an all-female panel, and it's going to take place in December. Please check back to our website for dates and locations. And with that, I would like to introduce you to one of our partners, Dorothy Kozlowski. Dr. Kozlowski is a professor of biological sciences and teaches neuroscience at DePaul University. Among many different awards and acknowledgements and recognitions, I picked one. She received the Educator of the Year award from the Faculty for Undergraduate Neuroscience, which is an international organization. Dorothy?
Thank you, Christina, for that wonderful introduction. So, as the president of the Chicago Society for Neuroscience, I'm very grateful to have been able to partner with C2ST. One of our mission statements, and I'm going to actually read this verbatim, which is why I brought this paper up here, is to disseminate to the general public cutting-edge research advancements in neuroscience-related disciplines. And to be honest with you, there's really nothing more cutting edge than the information you're going to hear about tonight. The BRAIN Initiative is the cutting edge of what neuroscience research is setting out to do. So I hope, on behalf of the Society, I'd like to welcome you. I'd like to thank you for coming to this wonderful program where you're going to learn about the brain and in turn learn about yourselves, because learning about neuroscience in the brain, really it's a little egotistical. We just want to know about ourselves. So enjoy. Oh. I'm supposed to introduce someone. Sorry about that. Norm Peterson, who is currently at the Argonne National Laboratory. He will play a role as our moderator tonight, and unfortunately I was told to introduce him at the last minute, so he's going to tell you more about himself and how wonderful he is. So, thank you, Norm.
Thanks, Dorothy and Christina, and also Alan for your leadership. Alan, as Christina mentioned, was a director of Argonne National Laboratory, and I really owe my career at Argonne to Alan because he was on a Governor's Commission on Science and Technology that I directed when I was with the state of Illinois, and Alan Treesheim hired me into Argonne, and I remember the first conversation I had with him about that was, what do you expect me to do at Argonne? So that was over 25 years ago. So thank you, Alan and Christina and Dorothy. Welcome tonight to What is the Brain Initiative? Tonight's topic is BRAIN, the Brain Initiative, which is spelled out, the Brain Research Through Advancing Innovative Neurotechnologies Initiative. It's an ambitious, relatively new program that was initiated recently by the National Institutes of Health. We're here today to learn more about it, why it's important, what neurological problems does this undertaking hope to solve. Today, 3 experts will be discussing their pioneering work and discuss how they hope to tackle some of brain's biggest mysteries. Last year, the National Institutes of Health budget for the Brain Initiative was $46 million. This year's projected budget is $81 million. NIH is also joined this year by a number of other federal agencies who are being involved in the Brain Initiative, including Food and Drug Administration, National Science Foundation, the Defense Advanced Research Project Agency, and from our perspective, the Department of Energy. Private corporations, foundations, universities, patient advocacy groups bring total annual investments in the Brain Initiative to over $300 million. Understanding the structure and function of the human brain is a Herculean task. The brain's a maze of 86 billion neurons with over 100 trillion connections between them. Mapping the brain would be a difficult enough task if the human brain was static. But it's not. It's ever-changing, varying not only from person to person, but within an individual, day to day. Ultimately, the Brain Initiative goal is to create a map of a dynamic living brain. While this project has been compared to the Human Genome Project, it is far larger in scope and the sheer amount of data collection analysis that is needed. Scientists eventually hope to gain a better understanding of the brain function, and with it, better insight into neurological and psychological disorders such as epilepsy, Parkinson, Alzheimer's disease, schizophrenia, bipolar disorder, and traumatic brain injury. These conditions affect millions of Americans and hundreds of millions worldwide. Better treatments for neurological conditions could be better developed with a better understanding of the brain. Argonne National Laboratory is pleased to partner with the Chicago Society for Neuroscience and C2ST on this program. I'd like to introduce our speakers this evening in order of their appearance. First speaker will be Dr. John Monsell. I believe John is the only Chicago scientist to have received direct funding from the Brain Initiative so far. He's the Alfred D. Lasker Professor of Neurobiology at the University of Chicago, who also serves as the director of the Grossman Institute for Neuroscience, Quantitative Biology and Human Behavior. He studies how the brain processes visual signals from the eyes, with special emphasis on how that processing varies depending on the task being performed. He received his PhD in biology from the California Institute of Technology. John will be followed by Dr. Bobby Katsuri, who is a neuroscience researcher and a recent addition to the staff of Argonne. He's also an assistant professor in the Department of Neurobiology at the University of Chicago. He has an MD from Washington University School of Medicine in St. Louis, and a doctorate in philosophy from Oxford University, where he studied as a Rhodes Scholar. As a postdoctoral researcher in the Lichtman Lab, Dr. Katsuri developed an automated approach to large volume serial electron microscopy, or connectomics. Katsuri Lab continues to innovate new approaches to electron microscope-based connectomics reconstructions, including making samples more amenable to automatic segmentation and combining proteomic and genomic approaches to electron microscopy. We're also now exploring the use of high-energy X-rays from synchrotron sources, like the advanced photon source at Argonne, for mapping brains in their entirety. Our third speaker this evening is Dr. Tom Masick, who's the Scientific Director of Clinical Sciences at the Takeda Development Center Americas. For the last 16 years, his primary focus has been on clinical development of new medications for the treatment of psychiatric and neurological disorders. He also holds a Doctor of Pharmacy degree from the University of Michigan and a PhD from Emory University. Following the presentations by the three speakers, we'll take questions from the audience at the microphones in either aisle. So at the conclusion of the presentations, the speakers will come up to the table. I'll moderate a discussion, and again, please hold your questions to the very end, and after the presentations, just walk to the aisle. So up first, John.
Very good. Well, thank you, Norm. And I'd like to thank the Chicago Council on Science and Technology for inviting me here this evening to talk with you about the BRAIN Initiative. We all know that the brain is amazing. Every thought, every emotion, every sensation, every movement that we make depends on the brain. And those accomplishments depend on an incredible amount of complexity. In round numbers, there are about 100 billion neurons in the brain. But that number is just the start of the complexity that's involved. Most cells in your body, I think it's on. Can you hear me in the back? Okay. Most cells in your body are little balls or cylinders, but the cells in your brain, the neurons, are different. Each of them has elaborate processes that grow and branch and spread in complicated ways. These are the dendrites that collect signals from cells around them, and the axons that take signals to other cells, sometimes very far away. Collectively, the connections, these sort of cellular wirings that you have on all these neurons, make up more than about 100,000 miles of cellular wiring in your brain. And the average cell connects with about 1,000 other cells in your brain. So they're about on the order of 100 trillion little cellular connections throughout the brain. It's an immensely complicated circuit. But it's even more complicated than that, because the cells are grouped in different ways, in different clusters with different connections and different sorts of organization. Everywhere you look in the brain, you'll see structure and patterns. If you look at this cross-section through a human brain, you can see the cerebellum, parts of the cerebral cortex, and the brainstem. But if you look closer and start looking under a microscope, you'll see that every small part of this structure has a different organization and a different group of cells. And there are literally hundreds and hundreds of different structures within the brain, each with their own function. We know in detail the function of some of these, but actually just a few. For most of them, we really have no clear statement about what those structures are doing. So the brain is just an immensely complicated sort of organ. If you wanted to understand how the liver worked, what you'd need to know is all of the molecular mechanisms, what they do, where they operate, and the cellular mechanisms that handle those molecular mechanisms. And then you'd be pretty much done. You'd have a reasonable good understanding of what the liver does for the organism. In the brain, you've more or less just started. The cellular mechanisms, the contacts between cells, are extremely complicated, more than what you find in most typical cells. But groups of cells are then organized into microcircuits. Dozens or hundreds of cells collected together, communicating with each other, getting inputs from other sites, sending inputs out, and performing a specific computation. We have some understanding about those sorts of computations and what goes on there, but then we get to a level where we really know relatively little, where you have hundreds or thousands of these microcircuits grouped together into structures which collectively perform some function. And those are grouped into larger structures which collectively work together in a coordinated way to eventually get you up to the level of cognition and behavior. So in this region, we're sort of lost. There may be 2 or 3 levels in there. And this is the state of affairs with the brains. It's an incredibly challenging sort of problem for us to be tackling. but an incredibly important one. Some of the reasons we need to understand the brain. Tom Masick is going to be talking in just a few minutes about some of the clinical aspects of the Brain Initiative. I just want to make the point that I think most of you already know. There are millions and millions of Americans who are suffering with brain disorders, which are completely debilitating or robbing them of the opportunity to live a full life. There are estimates that mental illness costs the world about $2.5 trillion every year through lost productivity and through treatments that are needed. And I will just say that the situation is only going to get worse as the lifespan continues to increase over time, because many of these diseases just simply appear with sort of menacingly regular intervals as people get older. Roughly half the population over the age of 85 suffers from Alzheimer's. Another reason we need to study the brain is that we can learn a lot. We have advanced technologies, but the brain does things that we would really like to be able to do. There's been great progress in artificial intelligence and robotics, but the fact of the matter is you can still literally run circles around the most advanced general-purpose robots in the world. I like showing this video because it's funny, but it's really not fair. The fact of the matter is computers have been better than people at chess for decades. And within a matter of years, computers will be better at driving cars or trucks than any person. But the fact of the matter is they are still rather graceless. They don't have the capabilities that simple animals have in terms of fluid motion. And they certainly don't have the capacity for sort of cognition and thought and creativity that humans do. Their computations are based on entirely different sorts of processes that occur in the brain, and we would like to understand what's going on in the brain in order to understand those processes. And finally, I just have to mention that it isn't the brain, it's us. Understanding the brain is probably going to tell us more about what it means to be human than any other venture going forward. So, for all of these reasons, we have the Brain Initiative, brain research through advancing innovative neurotechnologies. It's a collection of private and public funding, which is enabling hundreds of teams of scientists to develop and advance new technologies and put them to use to try and understand what's going on in the brain. Why now, you might ask yourself? Well, the chief reason is that during the past 5 to 10 years, there's actually been a flood of new technologies that have really given scientists the power to do experiments that make you feel like we've sort of got power that's commensurate with the problem that we're facing. The Human Genome Project spun off all sorts of new genetic technologies, which let us do incredible new experiments that we could only have dreamed about 20 years ago. Advances in computer power have really been phenomenal, not just the capacity to hold data, but the computational capacity that allows us to run experiments in real time that we couldn't do before, collect the data in real time, analyze it and visualize it in ways that just weren't possible a short time ago. And optical methods also turn out to be very important in biology. The sort of first weapon of choice is to see what's going on. And there have been some amazing accomplishments in advancing optical methods. And generally, by combining all three of these sorts of technologies together, we're able to do experiments that are truly remarkable. And what I'd like to do is just give you a sampling of some of the approaches that are now available and the methods that have come online to allow neuroscientists and others to begin this project of trying to understand how the brain works. And some of the questions they address are remarkably straightforward. They're exactly what you'd want to know to reverse engineer something and understand how it works. These tools are letting us get what's essentially a parts list. How many different types of cells are there in the brain? How many different molecular machines are there within a cell? Those things can now be approached. Wiring diagrams are critical. The brain is a big electrical circuit in many ways, and until we know how it's connected and the principles and patterns on which it's built, we won't be understanding what it's done. And there are now a variety of techniques that are giving us incredibly detailed information about the connectivity in the brain. And finally, we're in a position now where we can ask questions about function and dynamics, how the actual operations occur, how the brain does its job that we really couldn't approach before. I'd like to start with the idea of just getting a parts list. Some of this is perfectly straightforward. Different cells have different jobs, and they do it by using different molecular machines, which they produce by activating different genes. And so by looking at the sets of genes that different cells have inside them, we can actually distinguish different classes of cells. Cells that might look identical, in fact, have very different genetic expression and are doing different jobs and represent different classes of neurons. So having the ability to probe the cells and ask them what they are doing with their genes allows us to identify different classes of cells, ask where they are, how they're connected, and what sort of operations that they're performing. There are other techniques which allow us to look at cells by just making the brain transparent and looking inside it. So you can now take a chunk of brain from a mouse, treat it chemically to make it transparent, make one class of cell express a dye so that they can be visualized, and then you can look through this entire volume to see how they're laid out, which structures they're found in, which other cells they might be next to, and what sort of functions they might be performing based on that distribution. Microscopy has just gone to amazing levels. Traditionally, the limit in microscopy using light microscopy was the diffraction limit of light. We've now got methods that allow us to go past that, where we can actually look at individual molecules and follow their progression within cells. What you're seeing here is actually the growth and retraction of individual molecular filaments within living cells. So we can actually look how molecules and their cargo and their interactions are occurring within individual cells in real time as it occurs. Again, techniques that really weren't possible 10 years ago. In terms of the wiring diagram, there are a number of different approaches that are making important progress possible. In the human brain, it's now possible to take an ordinary subject and in a completely non-invasive way using magnetic resonance imaging, and in particular diffusion tract imaging, you can work out where the major connections, the sort of connective highways are inside that brain. Which parts of the cerebral cortex connect to which parts of the thalamus, which parts don't. Which groups of areas are working together and interconnected as a separate system, and which ones are combined into a single set of a single system. This gives you resolution at the sort of human brain level. Excuse me. There are other techniques that allow you to look at a much finer scale. In fact, one of them uses rabies virus. The reason rabies is so lethal is that once it gets into a neuron, it has mechanisms that allow it to get into all the neurons that connect to that neuron, and then all the neurons that connect to those neurons, so that essentially within a short period, you have all the cells in the brain infected with the virus. With genetic engineering, you can change a rabies virus, so it can only do that trick once. So that if you put it into a cell, it will go into all the cells connected to that cell and then stop. That gives you a way of finding out all the cells that are connected to any one cell that you've identified. It gives you the local circuitry and gives you the ability to try and infer what sort of computation that one cell is doing based on the cells that are giving it input. Great trick through genetic engineering. And then at an even finer scale, it's possible to do reconstructions that let you see every process, every connection in complete detail. Bobby Castori will be speaking in just a moment about this sort of approach, which allows you to look in small volumes to get every detail about the cellular connectivity that's going on in that region. Functionally, many of you will know that it's been possible to monitor the activity throughout a human brain non-invasively using functional magnetic resonance imaging. So for example, if you have a person in the scanner and have them do a visual task, you can see that the activity in the part of cerebral cortex that represents vision goes up, and the activity in the part of cerebral cortex that does hearing goes down. This is great and has provided a lot of insights into the organization of the human brain, but the problem is it's relatively low resolution. These smallest dots are spanning hundreds of thousands or 1 million neurons. We'd really like to have information at the level of individual cells. It's also relatively poor temporal resolution. You can only get one picture every few seconds, which is much longer than it takes to make a decision or implement an action. But we now have techniques in animals where we can begin to monitor the activity throughout a brain at the level of individual cells. What you're looking at here is some work done by some scientists who monitored the activity throughout the brain of a larval zebrafish, a growing zebrafish. In this case, Each of these flashes that you see, the individual dots, are individual neurons within the fish's brain. They used a technique where they got the cells to express proteins that allowed them to produce a visual signal every time they were electrically active. And then by monitoring those visual signals, they could watch the activity of 80,000 cells, virtually every cell in this animal's brain, on a time scale approaching the level at which behaviors are occurring. So effectively, you can just watch a brain in action and see little tiny fish thoughts flashing around in here. So these techniques are still limited in terms of the number of cells that can be monitored and the time scale over which it can be done. But with these sorts of approaches, you can now begin to ask questions about what happens when you introduce a visual stimulus and a motor action occurs. What are the cells engaged and what pathways are important? And the final approach I'd like to mention is that in addition to using light to monitor the activity of the brain, you can use light to change the activity of the brain. Using genetic tools again, you can get cells to become light sensitive in ways that will cause them to change their electrical activity when you shine a light on them. So for example, if a cell is silent, you can make it become active and start generating voltage pulses by shining a blue light. Or if it is active, you can make it become silent by shining a yellow light. This is critical because this allows you now to do perturbations and test hypotheses. If you now have an idea about what a circuit is doing, what a cell is contributing to a particular behavior, you can go in and actually manipulate the activity in that cell or in that circuit and test your hypothesis about what that behavior is doing. So to wrap up, I'd just like to say that it's an incredible time in neuroscience. It's wonderful to have this support, and there's going to be just a wash of new observations and new understanding coming out in neuroscience. I'd like to close with just one note of caution, though, about the Brain Initiative, and that is that it's frequently compared to a moonshot or the Human Genome Project. That's apt in some ways because it is a major commitment to a big scientific project involving interdisciplinary teams of scientists with a specific goal in mind. But it's different in a pretty critical way. In both of these cases, the goal was completely clear-cut. We knew exactly where the moon was, we knew just what the payload was, and it was a matter of engineering that to get someone up there and back. Similarly with the genome, the endpoint was clear. We knew exactly how much DNA there was in the genome. It was a matter of figuring out how to get the sequences out. The Brain Initiative is more a matter of discovery. The endpoint is to understand the brain, but we don't know how far away that is. It's possible that all the concepts and understanding will come tumbling out over the next 5 to 10 years, and we'll know how the brain works. But we really could be facing a much longer period than that before we come to a complete understanding. What I can guarantee is there's going to be a lot of exciting new findings coming out in the next few years. So thank you.
Thank you. As Norm introduced me, my name is Bobby Kasturi. I'm a neuroscience researcher at Argonne National Labs. I think perhaps the very first neuroscience researcher at Argonne National Labs, and I'll speak a little bit about that later on in the talk. I'm also an assistant professor adjunct in the Department of Neurobiology at the University of Chicago. And before I start, I just wanted to thank John. That was a really good, I think, general introduction to Brain Initiative and to sort of the technologies that are driving the Brain Initiative. For my part of the talk, I think I'm going to drill down on one of the very specific technologies that John described and tell you a little bit about the advances that we're making. But before I get to that, as I was thinking last night about how to start this talk, one of the things that occurred to me is that although the brain has all of these various senses, smelling, touching, hearing, tasting, and seeing, When we study the brain, we almost predominantly only use one of these senses. We almost exclusively use our eyes. Although we don't have to, it just seems to be historically, perhaps left over from the creation of books by Gutenberg, that we don't actually smell the brain to try to figure out how it works. And we very rarely taste the brain to figure out how it works. But I think in reality, a lot of the tools that we develop is so that we can actually visualize the brain. That once we actually see something, that we're able to actually believe it. And when I think about it, I think part of what the Brain Initiative is trying to achieve is to give us new ways to look at the brain. And I think the reason the Brain Initiative emphasizes this, as John so aptly demonstrated, is that every time we come up with a new way to look at the brain, we find a new surprise. And it's really these surprises that push the field forward. And in fact, as I thought a little bit more about this, I realized that there was actually a great philosopher of the Brain Initiative who I think summed it up best, and this is him. His name is Yogi Berra, and I apologize ahead of time for the New York Times, the New York Yankees cap, but I think this is right. You can observe a lot just by watching. And I think this is true not only in neuroscience, but in lots of other fields. The field that I feel the closest to is astronomy or astrophysics. And if you think about these scientists, they never really get the opportunity to do experiments at all, because the things that they're interested in are so far away and so massive that what they have learned is to infer rules for how the universe works just by watching, just by looking. And I'd like to make the argument, which I'd love to get feedback on, that right now, we're at the stage of what we know about the brain, that before we actually think any theories about it, it might be best to actually just watch it for a little while. And to give you a very specific example about how technology and watching can lead to great insights, I noticed that on the cover entrance, there's a picture of a brain produced by what I consider is probably the Albert Einstein of brains, and that's a gentleman called Santiago Ramon y Cajal. And I just want to go through this picture really quick together so we're all on the same page about how brains work. What's interesting about Santiago Ramon y Cajal is he used two technological advances available to him at the time he did his work, which is somewhere around the early 20th century, from about 1900 to say 1910 or 1911. And those two technological innovations were, one, the use of the compound microscope. I suspect that very few people had looked at brains using the resolving ability of a compound microscope. That was his first technological achievement. The second, which you can sort of see here, is a very particular stain that actually wasn't invented by Santiago Ramon y Cajal. It was invented by his Italian colleague named Camillo Golgi. And what this stain allowed Cajal to do was to label just a very small percentage of neurons in the brain. Probably less than 1% of all the neurons in the brain were labeled by Santiago Ramon y Cajal. And then what he did was he just looked inside microscopes, and of course there were no cameras at the time. He just drew the pictures that he saw. And let me go through one of those pictures with you so you can understand. So again, these are not photographs, these are drawings. And like all good drawings, Santiago has a lot of whiteout in this, but there are actually three fundamental laws of brains that he discovered that you can almost see in this picture. The very first idea is called the neuronal doctrine, which is the idea that brains are actually made up of individual cells, that they're discrete and not completely connected. Prior to this, I think there was an idea that the brain was one big cell, one big electrically coupled cell. And Santiago, based on his observations, said no. And in this picture, you can actually see four individual neurons. 1, 2, 3, and 4. And he gives them different letters to imply that they are actually different cells. So that's the first law. The second law that Santiago figured out is called the law of dynamic polarization. And this John alluded to, but you can also see very clearly in this picture. Here is a neuron. This is the cell body of that neuron. Inside that neuron is the nucleus where the DNA is stored. But you can tell by looking at this neuron that it doesn't look the same in all directions. It's not symmetrical. In fact, this neuron has a large tree that sticks out of it, which is we call the dendritic tree or the dendrite. And then on the other side of it is a very long cable, which we call an axon. And so usually when I talk about Santiago, it gets a little narcissistic because I think up to this point, I could have kept up with him, right? I mean, if I had the tools, if I had the compound microscope and I had the staining by Camilo Golgi, I think I could have at least discovered what he discovered. But there's a third thing in this picture, which I really think is a step beyond, possibly true genius. And that, sadly for us, are these arrows, because there are no arrows in the brain. Too bad, too bad. But I think what was amazing is that not looking at living brains, not looking at dynamic brains, by only looking at a series of fixed time points, fixed images of a brain, Santiago was able to infer something truly remarkable, and that's the direction of information transfer in the brain. So for example, he shows us, here is the axon of a cell labeled E, and it's transferring information, the cell labeled F, and it's transferring information into the dendrite of this cell labeled A, which then propagates that information down its axon into the dendrites of other neurons. Really the first example of what we call a neural network, how a series of neurons connected to each other can transfer information. And I'd like to emphasize again that this was done before we knew anything about the functional properties of neurons, that they fire electrical potentials, that they can be dynamic, that they can change, and I find it remarkable. The other thing I find remarkable, and I've stolen this title from one of my favorite authors, Marquez, is that given about 110 years, our idea of how neurons connect with each other hasn't changed significantly over this period. We still imagine neurons, this is from a review that I took about 2 years ago, we still imagine neurons making these beautiful paths and connecting to very specific cells. And what's incredible about this is that it's probably wrong. And the reason that it's wrong is that the stains that Santiago used and that we continue to use to this day only give us about what 1% of all the cells are actually doing. And to actually get all 99% in the same animal, it turns out that that's beyond our ability to do with light microscopes. Why? Because as John mentioned, we have something like 1 trillion synapses in our brain. And the only way to fit 1 trillion of anything in a volume this big is to make each element so small that it's actually below the wavelength of light. So this is still hard for me to think about, but if you can imagine light, there are actually structures inside the brain which are smaller than that. And therefore, you cannot use light microscopy to really get all of the details. So now I'm going to move away from this and talk about the work that I do, which is to use another modality, which is called electron microscopy, which allows us to use very high resolution images to map the brain. I just want to make sure we're all on the same page. So before we do that, let me just tell you how electron microscopy works, and then we can actually get to the data. What really happens is we take brains. Here it's a cute rodent, but it actually works for anything from humans all the way to octopuses. We take those brains out, and we have to stain them. We have to stain them with heavy metals because electrons get contrast, unlike light where you have fluorescent molecules, they get contrast with heavy metals. So the heavier the metal, the more likely you are to deflect the path of an electron, and the more likely I am to see that deflection in an image. So we often stain our samples with osmium and uranium and lead. And then the last thing we have to do is we have to embed these samples in plastic. Why would we do something crazy like that? Well, because it turns out that because there's a trade-off in everything in life, the trade-off with electrons is that although they give you very good resolution, they don't penetrate very deep into your sample. So that's the trade-off. So how do we compensate for that? We compensate for that by taking these brains, embedding them in plastic, and cutting them very thin. You can't cut a brain naturally very thin. It's like cutting wet spaghetti. And so it's very hard to get very thin, precise, deliberate slices from it. So by embedding this in plastic, you're able to then use something called a diamond knife to cut very thin sections off this. Now, none of this we invented. This has been around for decades. What we did was automate this process. So this all used to be done by human beings, but now it turns out that you can do all of this with machines. And I hope that makes sense to everybody. And what I'm going to show you on the next slide is what electron microscopic data of the brain actually looks like. And I'd like you to picture what the Cajal pictures look like while you're looking at this. So what you see here is a single EM section. Now remember, I'm cutting these brains, so what you're seeing is a two-dimensional representation of a three-dimensional object, and you're basically seeing the profiles or the contours of single neurons. And let me show you what a single neuron looks like in this, a piece of a single neuron looks like in this image. Here I can just tracing the membrane of a single neuron. Here I'm tracing the membrane of another neuron. And what's amazing, and I'll get back to this, is that once I show you this, usually it turns out that human beings are really good at doing this, at finding the membranes of different cells. But as you'll find out, computer algorithms are not as good at doing this, and we need them to be better because of about the size of the data. Now, the reason I pointed out this particular set, this particular neuron right here, and this particular neuron right here, as you can probably tell from your seat, this neuron is filled with little circles. Now, these aren't circles because it's really just 2D. In 3D, these are called spheres. These are spheres, and we call them synaptic vesicles. They are what contain the transmitter that communicates between two neurons, the synapses that Cajal identified. And in fact, I know it's an axon because only axons contain synaptic vesicles. And it is making a connection or a synapse to this cell right here, which is the dendrite, if you remember from when I was talking about Cajal, of another neuron. Now, of course, we don't have just individual sections. What we actually do is collect a volume of electron microscopic data, and then we do a very simple task, which has taken me years. We find one neuron, and we just circle it from section to section to section. And once you can circle this neuron from section to section to section, you can, of course, build a three-dimensional model of what these two neurons look like. So that's pretty good. We got two. Turns out that John said that there are probably close to 100 billion, so I'd like to fill in the other 999 million. That's a lot. Now, before we can do that, as a kind of test case, what we decided to do was take a very small part of a mouse brain, which is already a very small part compared to a human brain, and find all of the parts elements, as John was describing, in that extremely small piece of brain. And that's what I'm going to show you in the next slide. So imagine that I had just taken a kind of a biopsy core, a very small biopsy core into a mouse brain and then reconstructed it. And I'm going to show you what's in a mouse brain that's probably smaller than a grain of sand. And it turns out that this is what brains look like. Not how Cajal had imagined them, and not how we had thought about them for over 110 years, that the brain is just packed with these cellular processes that extend over very large distances and make connections with each other. Now, luckily for us, based on data that's been collected for many years, we can take that huge mess and partition it out into things that I was telling you, axons and dendrites. And another subtlety about brains is that there are actually two kinds of cells in brains. There's cells that we call excitatory neurons, which when they transmit information are much more likely to make their targets fire, and something called inhibitory neurons, which when they transmit information are less likely to make their targets fire. And again, this is smaller than a grain of sand, but this is the level of complexity of a mouse, which of course, I suspect, has less sophisticated behavior than most of you in this room. So I can only imagine what it would be like to map an extremely large brain. Now, this was all done for me as a postdoc. One of the reasons I'm at Argonne is there's actually two reasons, which I'll talk about. The first is that Argonne has some of the largest computing resources available. And if you want to map a brain at this particular resolution, you're going to collect a lot of data. And I'll tell you at the end of the talk what it is that I have in terms of data sizes. And the second reason is they have a very special microscope. I'm not sure they consider it a microscope. It's called the Advanced Photon Source. It's a synchrotron. Costs, I suspect, about $2 billion. But I think of it as the coolest microscope in the world. And what it allows us to do is that, like anyone who's done medical imaging, X-rays, unlike electrons, you don't have to slice your sample to get your X-ray all the way through. And it might be possible, actually, to get the same resolution that I can get with electron microscopy, but with X-rays and minus the slicing. And so just for a few slides, just because it's new work and I'm proud of it, I thought I would show you a little bit of what that looks like. Yeah? So what you're seeing now in this next picture is another piece of brain here embedded in plastic, just as I described. You can actually see the border of the plastic here, but now using X-ray microscopes in conjunction with beamline scientists at Argonne, we're now able to map this brain at something like 600 nanometer resolution in minutes and without having to slice. And without having to slice means we can take this data, let me just show you what it looks like at a little bit higher resolution. We can take this data and start identifying things. Each of these little dots that's appearing and disappearing in this movie is a neuron or a cell in the brain. Each of these little white squiggles that's traveling is the blood vessels. So the brain requires not only neurons, but it requires blood vessels to sort of supply the energy. And with this, we're already starting to make maps of what brains look like. So here we took that data, found every one of those neurons and every one of those blood vessels, and segmented it automatically. In this particular experiment, you guys might have heard of the Jennifer Aniston neuron. We were looking for there, and there it is. But using this data, what I'm interested in is answering fundamental questions about brains that people ask me all the time. How many neurons are there in a brain? Well, the answer is we don't know. We have approximations. How do the number of neurons change across brains? The answer is we don't know. And now we might actually have technology that allows us to answer these basic questions. And I suspect without answering these basic questions, it'll be extremely difficult to get to the very sophisticated questions that we're all interested in. One of the last things I'd like to talk about is data size, because something that's new to neuroscience, it certainly didn't exist when I started as a graduate student, is that most neuroscience has become big data science, because it turns out that it's actually hard to try to figure out the brain without imagining things like collecting the activity of every neuron, or collecting the connections between every neuron, or counting all of the neurons. So what I'd like to leave you with is an idea of how much data it would actually take to map the things that I'm interested in. So for example, to map a mouse brain, which is an extremely small animal, will take us something like 450,000 terabytes, or 450 million gigabytes of data. And that's probably more information than has ever been collected about any specific topic in the history of the world. Let me give you another example. If you took all the books and all the movies and all the audio and pictures in the Library of Congress, it would only actually be a very small fraction of this. Now, that's a mouse brain. To actually do a human brain at this would be 1.3 billion terabytes, or something like 1.3 trillion gigabytes. Now, this is a tough number, but it's actually not that bad, because it's just about half of all the hard drives that exist today. So if I could just get all of that, then I could easily map this human brain. But I think that's the level of complexity that we're trying to address. And of course, this can't be done without significant advances in computer science. And what I think is interesting is not only what computer science can do for neuroscience, but really what neuroscience is going to do for computer science. So someone was asking me outside in the lobby, what do I think of the neural networks, the algorithms, the computer algorithms, programs that people use? And I suspect they're great, but they have nothing to do with neurons or their networks, because until very recently, we have had no idea what neuronal networks look like. So I suspect this will be a two-way road where they will help us to analyze the data, and based on the principles of brains that we discover, they will then get actually better algorithms. And I hope that this loop becomes tighter and tighter and faster, because if it doesn't, I'm going to be out of a job. And the very last thing I'd like to leave you with, and John mentioned this, he stole my thunder a little bit, but I still think this is a good idea. When I think about this, I get a little depressed. I won't lie to you, but it's not. But usually, what cheers me up is that this is really nothing compared to what we have done in the past. And what I'm going to leave you with is really one of my favorite quotes, and it was a quote done by John F. Kennedy at Rice University in 1962, where Kennedy announces our plan to go to the moon. And maybe we can just read this together, because I can't imagine certainly some of the presidential candidates now speaking like this. And here it is. We choose to go to the moon. We choose to go to the moon in this decade and do the other things, not because they're easy, but because they're hard. Because that goal will serve to organize and measure the best of our energies and skills. Because that challenge is one that we're willing to accept, unwilling to postpone, and one which we intend to win. And for me, the best part here is that we do these things because they're hard. Because when we fail, and of course we will fail at some level, projects that happen like this never happen like they're imagined. Another recent saying I heard is that when you go to war, the first thing that's lost is the plan. And so I suspect just like that, the first thing that we will lose in our kind of attempt to get the brain will be the plan. But that's not the important part. The important part is when we try something this hard, we'll be able to know what our limits are. And without knowing that, I suspect this is all just an interesting intellectual exercise. Thank you very much.
Thank you. It's a real pleasure to be here speaking to you tonight. I'd just like to take a moment to thank C2ST. The little that I've known about them over the last few months, I think their mission and objectives are very noble. And so to those in the audience, please consider, if, for each of the talks that you like, maybe make a small donation of $5. So I think most of us are on the hook for at least $10. This is unsolicited. But based on the cool pictures that the previous speakers have shown, I don't know if we're going to get to 15. I come to you today as a neuroscientist, a clinical neuroscientist, who's truly dedicated to the advancement of new medications for diseases. It's important to tell you that I speak today not on behalf of my company, and the opinions that I'll be expressing today are my own, as well as the images may not be reproduced without the permission of the commissioner of baseball. So I work primarily in psychiatry, and I think it's important that we first ground ourselves in understanding what is mental illness and the impact that it has on society. It affects their thinking, their mood, their ability to relate to others. Hundreds of millions of people worldwide are affected by by mental illness. I put some numbers here: 154 million from depression, 25 million from schizophrenia, 100 million from substance abuse disorders. And we can measure the costs of mental illness on society using something called disability-adjusted life years. And the World Health Organization has estimated that mental illness in itself accounts for 37% of all the of healthy years lost by non-communicable diseases. Depression in itself accounts for a third of all these disabilities. And the costs for treatment in 2010 were $2.5 trillion and projected to be $6 trillion by 2030. In comparison, 2009, the total health spending was $5.1 trillion globally. In mental illness, cardiovascular disease, diabetes, respiratory disease are also common comorbid diseases, and so the actual estimates of what we spend on treating mental illness is probably grossly underestimated. And we look at the individual mental illnesses, one like schizophrenia, which I have particular interest in. It affects 1% of the population. However, when we look at those dailies and the amount of impact, the percentage that it has, 1% of the population accounts for 8.4% of all the dailies, the disability-adjusted life years. I work in drug development in the pharmaceutical industry, and that's the process of bringing new drugs to patients through the process of drug discovery. We do research in animals, in microorganisms, and then the area which I work is in clinical trials and to determining whether they're safe and effective in humans. Preclinical research is that part where we're looking in animals to see if a promising target has potential activity in treating a disease. We don't usually know the safety of a compound. We don't know how it will be metabolized in humans. We don't know if it'll work in humans. So the preclinical development aspect of the pharmaceutical industry is dedicated to understanding in animals how drugs might help people. Clinical research is what we do to determine the efficacy and safety of these drugs. These might be prevention, treatment, diagnosis, relieving symptoms of disease, but it's different from clinical practice. Clinical research is trying to determine whether a treatment is safe and effective compared to clinical practice, which is the use of established treatments. Now, in what we do in clinical research is the pharmaceutical industry, it would be unethical to expose large numbers of patients to an unknown compound that may or may not be safe or may not be effective. So we do it in phases of research. The first phase being in a very few small number of patients to see how it's metabolized or tolerated. Phase 2 are early signs of efficacy as well as the safety of the compound. And then what we call phase 3 is where we're going into potentially thousands of patients to determine whether or not a drug is potentially effective. And the costs of doing this research are immense. I only show this not to, the pharmaceutical industry has been in the news a little bit in the last week. I'm not speaking at all to that. And what I'm speaking to is just to highlight the cost of developing new drugs. On the left, in the brownish color, you see the preclinical costs. And at each stage of development, there's attrition. So a drug might be found to be toxic in animals or not potentially effective. And at each stage, there's costs and there are attritions. Same with clinical research. When we go from small numbers of patients to larger numbers of patients, we may detect signals that are indicative that the drug is not safe or not work. The estimates, the most recent ones, of the out-of-pocket costs to the pharmaceutical industry is for each new compound is $873 million, and in capitalized costs is $1.7 billion for each new drug that comes to market. In CNS, it's particularly difficult. We don't understand the diseases as well as we think we do. The targets are not well identified, and all of the sophisticated work, even going back to Neurological, we don't really truly understand how the brain works. We also have somewhat variable diagnostic criteria. In mental illness in particular, it's not blood pressure or it's not blood sugar for diabetes. We're talking about subjective measures that are gleaned through a clinical clinical evaluation or interview with a patient. And also what we know also in mental illness is these aren't singular diseases. Schizophrenia is not a singular disease. There are different types, there are different presentations, disease course, and our fundamental understanding of a disease like schizophrenia is greatly lacking. In CNS and in CNS development, the really disheartening thing is that the failure rate is large. Of all of the drugs in CNS, the failure rate from 1993 to 2004, 1 in 10 failed. Or excuse me, 1 in 10 made it, compared to 1 in 6 for all other drugs. Since 2000, the approval rate of CNS drugs is declining, as well as it takes much longer to develop new treatments for mental illness and other CNS disorders. What this has led to in my industry is a number of companies are leaving. A number of companies are not devoting the resources to psychiatry or other neurological diseases. Here are some excerpts from Wall Street Journal, a quote from Thomas Insel noting AstraZeneca, GlaxoSmithKline, Sanofi Aventis, and Novartis exiting from neuroscience research and going to things that are considered less risky in development, like cancer. So there's a giant shift in our focus in finding new medications. Part of it, and one of the really sobering things about the history of drug development and CNS discovery, is most of them are serendipitous. For example, the first antipsychotic, chlorpromazine, was originally developed to be used in anesthesia to prevent shock. It was only later that it was noted that it controlled symptoms of psychosis and the term antipsychotic developed. Lithium salts, which are used to treat bipolar disorder in the 1850s, were originally used for the treatment of gout, and it wasn't until almost 100 years later that the relationship of lithium and the potential in bipolar disorder were realized. And then antidepressants, which I'll talk a bit more about. In the treatment of tuberculosis with a drug called isoniazid, it was noted that in patients, it seemed to help those who were most severely ill improve their mood. And this is where the first term, the antidepressant term, was coined. And it was not through some systematic understanding of the brain or disease. These were all by accident. In many cases, when I give this type of talk, when I talk about antipsychotics and even antidepressants, I don't believe truly there has been a fundamental advance in the last 50 years, because most of the drugs that we have, particularly in schizophrenia, all are fundamentally the same. Now I'm going to talk a little bit more about depression. And depression and diagnosing depression isn't as straightforward as hypertension or others. It's a collection of symptoms, not only sadness, but there are other collections of symptoms that, according to the diagnostic criteria that has been developed and used in the US to diagnose depression, are things like depressed mood, that's the easier one to, but loss of interest or pleasure, one of those two. Weight, changes in weight or appetite, either increases or decreases in sleep, either psychomotor retardation or agitation. So these are not really well defined, even in our current diagnostic criteria. And then we get to the issue of preclinical research and finding new drugs. How to tell if your rat is depressed. So it's impossible in rats to completely mimic those symptoms of depression that we observe in humans. We have developed paradigms to detect specific behavioral differences that are responsive to the effects of antidepressant treatment. And I'm now going to describe some of the most common ways we find antidepressants. One of the most common is called the forced swim test, and a rodent is put into some vessel of water, and the rodent doesn't want to drown. So there are 3 periods or 3 different types of activity that are noted: climbing, swimming, and immobility. And the immobility is thought to be representative of helplessness. What was noticed was that if you give an antidepressant, when you put them in this vessel of water the second time, the amount of time that they are immobile is lessened. So they're trying more. And this is how we screen for antidepressants in the pharmaceutical industry. One of the other most common is you take a rat or a mouse and you hang it up by its tail. And the concepts are very similar. It's the time to when they give up. It's either, in this case, the immobility or the swinging or curling behaviors. And what was noted in the tail suspension test is if you give an antidepressant, shown on the left, the time of immobility is lessened. So it seems like these rats and mice are trying a little more, either not to drown or to right themselves. Now, I mentioned that CNS hypertension, these are all subjective, especially in psychiatry, measures that we use that are subjective to clinical report, patient report. There aren't blood tests. There aren't definitive genetic markers. There are not really other surrogate markers that are indicative of treatment effectiveness in humans. And although that is a developing area, we are beginning to use techniques such as imaging and EEG to better understand either those populations that may or may not respond to a given treatment, or to identify subpopulations that may or may not respond differently to a different treatment. These are collectively sometimes called biomarkers. So through the BRAIN Initiative, and as a clinical neuroscientist, my hope, along with the really exciting science, is that we do develop a more comprehensive understanding of neuropsychiatric disorders in humans, which is one of the main goals of the BRAIN Initiative. Also, in parallel, it is hoped that this will provide better animal models for understanding our diseases and even screening for new drugs to treat them. And then also the identification of biomarkers that may help us or other surrogate measures that may help us to be indicative of treatment response. As it translates to us in the pharmaceutical industry and in the scientific, academic, and other scientific communities, is my hope is that this brings us an era of greater collaboration. It promotes the sharing of knowledge and information in what we would call in industry a pre-competitive space. That is a number of different scientists working together without the pressures of competition. New technologies that may be developed to either understand or the diagnosis of disease or measurement of therapeutic response. New targets that might be useful in the treatment of disease, as well as the neurocircuitry involved in disease. These biomarkers that I've talked about are so important because I mentioned how long it takes and how difficult it is to develop drugs in CNS. If we could reduce that attrition in drugs as they go through clinical development, if we can use biomarkers for better decision making, to make decisions earlier, to reduce ineffective medications, the exposure to patients, it allows us in the industry to better focus our resources on treatments that will be more promising and potentially successful, as well as these biomarkers may help us identify those subpopulations, either who may respond to treatment or maybe different flavors, as I call it, of the disease. I'm now going to take off my neuroscientist hat and I'm going to step into my tax and spend bleeding heart liberal mode. The estimates for this grant, for the BRAIN Initiative, for NIH funding, though it is a partnership of public and private funding over the 12 years, is $4.5 billion, a lot of money. And what I think was really interesting was one of the architects of the BRAIN Initiative put the cost in perspective for everyday Americans, for us as taxpayers, as the amount that's being invested is the equivalent to about 1 6-pack of beer for every American over the course of the BRAIN Initiative. I did the math using some just generic numbers of 320 million people, 4.5 billion years, $4.5 billion, about $14 a 6-pack. That's a pretty good 6-pack of beer. And as a connoisseur of beer, I want to put something else in perspective, though. Other governmental initiatives. The F-35 Joint Strike Fighter is supposed to be the most sophisticated warplane ever developed. The Pentagon plans to spend $391 billion on a little over 2,400 aircraft, each plane costing $160 million. From the most recent estimates over the course of the F-35 program, the costs are expected to exceed $1.5 trillion. Putting that into perspective, in beer, that $390 billion is a $1,200 6-pack, or 87 of those $14 6-packs. I think I would take the 87 from $47 6-pack. You can also put it into a different perspective. From the pharmaceutical industry and the amount that we spend developing new medications, it's 28 planes that are being dedicated to the BRAIN Initiative by the NIH, or about 5 new drugs that within the pharmaceutical industry we spend to develop. Thank you.
So initially, please join me in thanking the 3 panelists for just a wonderful set of presentations. So I had mentioned earlier, if you have questions, please go to either microphone and both sides of the aisle here. But I wanted to kind of tee that up, maybe a question to John and Bobby. In terms of the current technology tools, synchrotrons, electron microscopy centers, high-performance computing, how close are we to really reach the goals that you both have?
Not close at all. It depends on the particular goal. The idea is to get a map of the idea is to get a map. If the goal is to map a human brain at the level of every connection in a human brain, then we're not close. But as I was saying earlier, Sorry? Okay, is that better? Great, sorry. So then the answer's still not close. I feel like I've said it enough. Yeah. In fact, I'm going to let John go since I've said not close enough.
Well, I have to agree. If we go for just the raw numbers, we'll just be overwhelmed. The hope is that what we'll do is recognize the patterns and the organization and be able to make a synthesis so we won't have to actually do complete reconstructions of many human brains. But we do have the tools to make inroads in virtually every part of the brain, and with luck, that'll be enough for us to get the principles.
Great, thank you. Let's start on that side. If you can just state your name and an organizational affiliation.
Rob Klips, formerly with the University of Illinois at Chicago. Are there any strongly sex-related or sex-specific diseases, neurological diseases?
There are some. The ones that come to mind are the genetic ones, such as Fragile X, which occurs in men or boys, is the leading cause of genetic mental retardation. There are differences in sex differences in the overall incidence. Fibromyalgia, for one, is one that has a slightly higher incidence in women. But I hope that addresses your question. But there aren't a lot that are cut and dry, particularly as I come from psychiatry.
I might just add, for most of the questions that we're addressing in terms of brain organization and function, there have not been sex differences that have been noted. But I would add that there haven't been sex differences really studied as a specific research project. I will add that the National Institutes of Health are very sensitized to this question, and those of us who are now doing animal experiments or human experiments are required to record the sex of every subject that we use, and in our publications to actually split the results in between the sexes. Even if there is no difference, it has to be reported now in the hope that we may be able to pick up on sex differences and identify important information that comes that way.
Let's go with this side.
Thank you all for coming. My name is Christian. I'm a recent graduate at the University of Chicago. This question could probably be for any of you all. So a couple of you mentioned that there's kind of a gap between what we know from the top down, so kind of behavior and cognition, and from the bottom up, so like the cellular, molecular levels. Do any of you know if the Brain Initiative is tackling kind of that gap between what we see in behavior and psychiatric disorders and then like the neurocircuitry level, so just kind of somewhere in that middle?
I would say if you actually read the Brain Initiative report from the panel, which is a tome, a major document, one of the things they emphasize is how little we know about that span of organization. So each of those levels I referred to, molecular, cellular, microcircuitry, up to behavior, at each level, the subsequent level is coming out with emergent properties that just didn't exist at the earlier level. And the place where our understanding really drops out is when you get above the level of hundreds or perhaps 1,000 neurons. And there's a specific directive in the Brain Initiative to try and focus on how that coordinated behavior comes out from these circuits and try to relate it to disorders.
Chris?
Ralph Wanger, IIT. As you go up the evolutionary scale, can you tell how the things that make a brain human develop versus other animals, and in humans, how much of the brain that is?
So I don't have a particular answer for a particular brain region, but in general, it's thought that as you go up the evolutionary scale, what we call cortex becomes dominant and therefore is responsible for the kinds of decisions that humans make and sensations and thought. But I'd like to actually push the question in a different direction, which is one of the fascinating things for me is that what's different about humans
probably than any other animal, is that we spend a lot of time learning after we're born. As opposed to a fly, which is essentially born knowing how to fly, which is a very complicated task. If you think about navigating in three-dimensional space, that's a hard job that it's born basically knowing, but sadly, it will never learn anything else. And what I find interesting is that going up the evolutionary scale is a kind of slowing of development, that animals, particularly humans that spend lots of time learning, wind up progressing to much more sophisticated behaviors. And other animals that we consider intelligent, elephants, dolphins, whales, primates, also share this common motif of a very protracted developmental period where you spend a lot of time training your brain to accommodate to the very specific environment that it's in.
This side.
Hi, I kind of had a question on what you consider some of the greatest computational challenges that you face right now, storage aside. So obviously that's a huge problem, but also I'm just wondering what's interesting right now in terms of signal processing, computer vision, straight up computation in a quantum sense or otherwise. Kind of what are some of the other areas that are definitely of interest right now?
Well, I would say it's fair to say that the field is struggling a bit with how to analyze the masses of data that come in. So in just one example, we now have this capability I referred to record simultaneously with high temporal resolution from hundreds of neurons. And the question is, what are you going to do with that information? How are you going to pull a synthesis out of that sort of information? You could start by looking at whether cells have correlated activity, whether the firing of one cell always is followed by the firing of another, or if it's followed in a probabilistic way. And people do those sorts of approaches, but you can quickly get into very higher order statistics which become intractable mathematically. So we have a lot of theorists and mathematicians who are trying to work out methods for approaching that large volume of data where we can get guidance from the analysis instead of trying to come up with sort of after-the-fact hypotheses about how should we think about this set of spiking active neurons now that we've collected it. So, the challenge is conceptual and what frameworks we're going to try and put the data into. So there are issues, of course, about storing the data and getting access to the data and visualizing it, but part of the challenge is just getting the proper theory to work with.
That side. Name and affiliation, please.
Hi there, I'm Jeff Montgomery. I'm at UChicago. My question is about, you talked about how it's hard to, it's really easy for people to see cell membranes, for instance, but computers aren't good at it. Are there any methods or ways that you're trying to get people together to work on this to help develop those methods? And also on that same line of sort of sharing and working together, Dr. Masek, you mentioned how the pre-competitive space of academia is ideal for the development of preclinical pharmaceuticals. What sort of ways can we work together on that level when there's still the competition to publish and get the first results?
Yeah, so let me answer the part that I heard, and I might have gotten it wrong, so I apologize. The computer algorithms are not as good as human beings to sort of do image analysis, find the things in images that we care about. There are several groups that are trying to distribute this, crowdsource this, based on internet games. So I'd read once that, in fact, I still think this is true, that if 1% of the people that played Angry Birds wound up doing human brain analysis or EM analysis, we'd be done mapping the brain by now. So I think that's a significant opportunity. No, I think that's true, actually, based. So I think a significant opportunity to crowdsource this is to try to make it into a game. My particular version of this is to use high school students because they're very cheap labor and they're extremely motivated if you can teach them the right way. And I think that's another opportunity that neuroscience presents, is that we have lots of data that we have collected about brains, and there are lots of students interested in the world about brains, and we just need to match those two together. I'm not exactly sure if I answered your question, but okay.
I think I will also just make a comment that probably also doesn't answer your question, just because you mentioned the business about working together. And I don't think any of us really emphasize that neuroscience is arguably the most interdisciplinary of the sciences. And one of the things that I think all of us really like about it is we end up collaborating with scientists from almost across the spectrum. So in terms of vision and recognition, we talk to computer scientists who are doing machine learning and trying to develop these image segmentation mechanisms and are trying to do it in a way that relates to how it might be done biologically. We're talking to psychologists who have all the framework for understanding memory and attention and what sort of constructs people use when they're doing those sorts of tasks that we need in our behaving experiments. Physicists and engineers are the ones who are really helping us with the tools that are needed for improving our ability to get better resolution in our experiments and do things that we can't quite do yet. And then we're also talking to the geneticists, who are, of course, key for a lot of the manipulations that we see. And then obviously the clinicians, the people in neurology departments, in pediatrics departments, psychiatry departments. They're the ones who are really telling us what some of the main issues are and giving us guidance in that domain. So one of the nice things about neuroscience is we're always working with all of these different scientists trying to pull it all together.
On this side.
My name is Ed Waldridge. I'm a retired physicist. And my question is, how do you anticipate your very impressive studies of very small elements of the brain and with much data, how will that lead to an understanding of consciousness?
I think a lot of us got into neuroscience by thinking about consciousness. I think it's the one thing that we would all really like to tackle. And I think, as scientists, we recognize that consciousness is a construct. It comes out of the brain. It might be an illusion, but even illusions need explanations. And the trouble we face now is that no one has formulated things in a clear and precise enough way to allow us to do objective experiments on consciousness. You can dance around it in terms of whether people can report or see this thing and tell us that they were aware of it, but there really isn't a direct way yet to get at the notion of this subjective experience with an objective experiment. But I think, I won't speak for my colleagues, I think it's on our plate. It's a point we have to get to, and I do think that it's going to fall into place once we get to a clearer understanding about the various mechanisms that are supporting these decisions, these perceptions, and these actions that we make.
What side?
I'm Dr. Naveen Yadav from Midwestern University, Downers Grove. I have a question about the diffusion tensor imaging. So it is recommended to use the DTI in the brain injury patients. So what's your what's your comments on the DTI using in the brain injury or concussion patient? Because there's still controversy using the DTI in these type of patients or DTI patients to assess their damage in different areas rather than using CT or X-rays.
I get the irony here, but could you speak a little bit closer into the microphone? I'm sorry.
So I have a question about the DTI, diffusion tensor imaging. So it is recommended to use the DTI in the brain injury patients or concussion patient because it's good tool to assess their damage at the sensory level, at the axonal level. So what's your comment or experience about that? Like, because there's still a controversy, like we should use it, like we can assess the damage, but there are some studies which shows that DTI is not useful, like to assess the damage in these patients.
Well, I'm at the very opposite end of the resolution scale relative to DTI. So I look at things at the nanometer resolution. So I'm slightly biased by what I'm about to say. But what I'm about to say is that it's not clear to me that there's sufficient resolution using DTI to find the proximate cause of, and I think you were talking about concussion, and traumatic brain injury. I've heard that these might be things that are injuries that are distributed over the brain, that there might not be a focal center of it. And if they are distributed, it might be hard to actually find the particular location that is the cause of a concussion or traumatic brain injury. But again, I'm biased.
Thank you.
Yes, sir.
Yeah, I was hoping to find here that you will be able to lead us into have a better quality of leadership, people with empathy and with understanding of the universe we live in. It's very obvious that we don't have that in our leaders and potential leaders, specifically the lack of empathy that is so obvious into these people who pretend to be leaders. Is anything in your studies that could somehow find out whether this is present or not present, or why is or isn't present in the people?
I think we'll probably solve consciousness before we figure out a way to get our leaders to be empathetic. I think these are deep questions about how the brain works. Emotions have been very difficult for people to address, again, because we sort of lack the correct constructs. But these are all the things that we want to work towards understanding. But I think that is one of the aspects I would put as sort of the distant goals. It will probably be a long time before we understand why it is that one person has empathy and another person does not.
I think also part of your question was empathy as it relates to patients with disease. And you also mentioned leadership, incorporating, if I understood correctly, empathy. For myself, I will just say it is the empathy that I have for those patients who suffer from severe mental illness is why I have committed my life to this work.
Over on this side again.
I have to cut us off, actually. We are out of time, so thank you all for your questions.
Why don't we take one more question, Chris? Go ahead, please.
I'm kind of pushy. Okay, I suffer from depression, but I read the article in the National Geographic 2 years ago about the brain, and you did talk about some of the exact same things that the article discussed, and also have they done research for children with autism. I'm a retired teacher, so it's like I think about the two extremes, the autistic children, and it's curious to me what goes on inside of their brains. You know, they seem as they're in another world. And then also Alzheimer's, you know, have they looked into, because that's my biggest fear is like, you know, getting 80 and then all of a sudden I'm like, you know, don't know where I am. So are they, you know, working on those two things, autism and
Right.
and
So to autism, I'll tell you that neurodevelopmental disorders are a huge part of the current focus of CNS research, and a number of other companies like Roche and Novartis have made significant investments into understanding autism and potentially treatments for autism. To the Alzheimer's disease, the amount of money that has been spent in the last 3 to 5 years in just Alzheimer's disease trials, I would estimate is greater than that $4.5 billion that the NIH has committed to the Brain Initiative. We are all dedicated to, especially for all of these disorders, finding new treatments. The sad fact is we've had a lot of those late-stage failures where in mice and rats we're able to have dramatic improvement in Alzheimer's-like symptoms or Alzheimer's-like pathology, but unfortunately it has not translated to humans. And I'd be happy to talk more with you about after the session.
So thank you, C2ST and Dorothy and the panel. Thanks, Bobby.